Something else to note here about what's happening in the large subunit of the ribosome. The large subunit is where catalysis is happening. And it turns out that the active site, the place in the large subunit where the peptide bond is being built, is made entirely of rRNA. This is new for us. Normally, we expect catalysis to be performed by an enzyme. And remember, by definition, an enzyme is made out of protein. This is different. What's catalytic here is not protein, it's rRNA. So it needs a new name. We refer to this catalytic rRNA as a ribozyme, not an enzyme. Ribosomes are likely one of the most ancient molecules inside living cells. Remember, they are found in all three domains. That means that ribosomes were present at the beginning of cellular life on this planet. It makes sense that they have a more ancient catalytic system in them, based on rRNA instead of protein. The other thing for us to note here is that the process of elongation requires energy. And interestingly, it doesn't come in the form of ATP, it comes in the form of GTP. So just as I said a minute ago, that ribosomes are an ancient molecule, it's probably no surprise to us that they are using the more ancient form of energy currency, which is GTP. Elongation requires assistance from additional sort of supplemental proteins. So just like we saw initiation factors helping out during the initiation phase, there are elongation factors helping out during elongation. They're helping stabilize this process, in other words. Remember that as the ribosome translocates, as it travels down the length of the mRNA during translation, other ribosomes are going to come in and start the process. A second small subunit will come in through initiation and guide a second large subunit in, and a second polypeptide will be built from this mRNA. And then another ribosome will come in, and another. In fact, ribosomes will continue to bind to each mRNA and translate it as long as it exists, until it is degraded in the cell. And remember, when we see an mRNA with several ribosomes sitting on it doing translation, we give that a name. We call that a polyribosome. And that's what we're looking at on this slide. There's a drawing of it down here on the bottom, and there's an actual electron micrograph up at the top. So let's take a look. The yellow mRNA strand is this very thin line that you can see through the picture. The circular, sort of gold-colored structures, those are ribosomes. And the green that you see, those are polypeptides. So a ribosome comes on to the mRNA at the start codon and begins to translate. And you can see that this ribosome, which is sitting maybe at the second or third codon, has a very short green strand coming out of it. If we check back a few minutes later, this ribosome has moved. Maybe it's all the way down here now. And its green polypeptide has gotten a lot longer because it's traveled down that many codons, and it has added that many amino acids to the chain. And if we come back a few minutes later, it's probably over here. And it has an even longer green chain on it. But this ribosome that started here, as it moves this way, it's going to get joined by other ribosomes. another one will come in and begin and move, and another one will come in and begin, and another one will come in and begin. So that if we take a peek at this mRNA at any one moment, it's going to look like this. It's going to have all these ribosomes on it. And depending on where they are in the process, they have longer and longer and longer polypeptides coming out of them. This is a polyribosome structure, and you can see it as it exists in nature right here. Brings us to the last phase of translation, the termination phase. So the question is, what is it that tells a ribosome to stop translating an mRNA? And the answer is the stop codon. Once the ribosome translocates onto a stop codon, in other words, once it reaches the part of the mRNA where the stop codon is exposed, a very special tRNA is going to come into the A site. And we call these tRNAs release factors. So you have to imagine that the ribosome has translocated to the next codon. That codon happens to be a stop codon. And recall from the genetic code, there are actually 3 different stop codons. So the only tRNA that will have the correct anticodon for these stop codons are release factor tRNAs. Release factor tRNAs look just like all the other tRNAs, except they don't carry an amino acid. So the stop codon is reached, a release factor comes into the A site with no amino acid on it. The ribosome tries to build a peptide bond and can't because there's no amino acid. And that will cause the process to stop. The small and large subunits will release from the mRNA, and the polypeptide will be released from the large subunit. And that's what they're showing us here. On the left-hand side of the diagram, you can see a ribosome, small subunit, large subunit. You can see the mRNA sitting between them. You can see the growing polypeptide chain of amino acids right here. You can see that there are 3 tRNA in the 3 sites of the large subunit. And what has happened is the ribosome has reached a stop codon. This one happens to be UAG. So the only tRNA that can come in here and bind to this stop codon is a release factor tRNA. It has no amino acid on it. So when the large subunit tries to build a peptide bond between this last amino acid and this missing amino acid, it can't. And that's going to cause the whole thing to stop. It's going to cause the 2 subunits to dissociate from the mRNA and the polypeptide to be released from the large subunit, and translation ends. Now, once the polypeptide is released from the large subunit, it's not yet a protein. It's not yet a mature protein. We talked a little bit about that language earlier in the semester. In order for it to be a true protein, the polypeptide has to be folded into its own unique 3-dimensional shape. Now remember, in living cells, polypeptides are long chains of amino acids, usually hundreds to thousands of amino acids. And the way they fold is going to depend on which amino acids are next to each other, what the sequence of amino acids is, in other words. Because the R groups that are extending off of each amino acid are going to interact with each other. And depending on whether or not they are polar or non-polar. We're going to see things like hydrogen bonding and van der Waals interactions going on. And we're going to see that molecule fold into, remember, alpha helices and beta sheets, and then the final three-dimensional functional protein. Now, as you might imagine, when you have a long chain of amino acids, again, hundreds to thousands, there are actually several ways that that chain can fold. It's not just one folding possibility, it's many folding possibilities. And it's critically important that that polypeptide fold into the one three-dimensional shape that that protein needs to be in order to do its job. And that's where a very interesting type of molecule comes in called a molecular chaperone. And just like the chaperones that used to attend the dances that you went to in high school, these chaperones are there to watch over the folding process. So each step of this folding of the polypeptide is guided, and it's guided by these molecular chaperones. And that's going to assure that that polypeptide folds into the one correct shape that it needs to be in order to do its final function.